topic saying that at this stage, the current portfolio for the treatment of multiple sclerosis with stem cells include hematopoietic stem cells, and this treatment is based on the ability of mobilize hematopoietic stem cells from the bone marrow, and following severe chemotherapy that leads to the ablation of the immune system will then result in the infusion of these cells with the aim of reconstitute a healthy immune system, at the same time eradicate autoimmunity, and possibly reset the immune system toward a more tolerogenic one. Other cells involved in or possibly involved in the treatment of multiple sclerosis are neuroprecursor cells, and many of you know the work of Gianvito Martina and Stefano Pluchina at San Raffaele Hospital, who demonstrated that these cells that can be obtained from the brain, or particularly the particular areas of the brain, can be expanded in vitro and injected intravenously, leading to, at least in animal models, to tissue repair through different mechanisms, which include transdifferentiation and neuroprotection, induction of local neurogenesis, and immunomodulation. But what I will going to talk about are these cells, namely mesenchymal stem cells, that can be isolated from different tissues, mainly from the bone marrow and adipose tissues, but also from almost any connective tissues. These cells, at least those that are isolated from the bone marrow, are known to play a major role in the maintenance of the hematopoietic stem cell niche through the interactions with hematopoietic stem cells. I invite you to, if you're interested, to read this fantastic work from Paul Frenet, who published in August last year a detailed analysis of the interactions between the mesenchymal and hematopoietic stem cells within the niche, where, as I said, the larger part of the stem cells population is composed by hematopoietic stem cells, and a very limited amount of cells, less than 0.001%, are cells with from the mesodermal lineage, which are, as I said, named mesenchymal stem cells. What's the rationale for using these cells for the treatment of autoimmunity? There's no doubt that everybody working on in the stem cell field has moved in with the idea that this cell could provide tissue repair through transdifferentiation mechanisms, meaning regenerate the tissue through their ability to generate new cells, potentially also from other embryonic lineages. But as you will see, more recent data have shown that these cells can indeed play an important role due to their ability to interact with immune cells and provide bystander effects, which leads to the release of different molecules with trophic, anti-apoptotic, antioxidant effects, and potentially also with the ability of mobilize and local precursors neural cells. This is extremely bright. Nevertheless, this slide shows you that MSCs have the ability to become neurons. This is a study published by Bruno Bonetti in Verona, who nicely demonstrated, although it wasn't the first, but I think was a very nice demonstration, that these cells can acquire a neuronal phenotype, and it was able also to show that MSCs, MSCs-derived neural cells can also have an electric activity. Nevertheless, as you will see later on, I have to admit that the experimental evidence that transdifferentiation into neural cells occurs in vitro as well is quite limited and very controversial, and I personally do not believe it. One of the, as I said before, one of the mechanisms that could, trigger our curiosity about the possibility of using MSCs for neurodegenerative disease derives also from the well-known ability of MSCs to promote survival of neural cells. In this paper published by the group of Niels Golding, it was stated that, and this is just an example, MSCs can secrete neurotrophic factors, in this case is BDNF, which promotes, as I said, neuronal survival. One of the most intriguing effect that has been provided in the literature by this group, which is a group located in the United States, is Darwin Sprokop's group in Texas, was the demonstration that when you inject MSCs, in this case, in this cartoon, MSCs are in pink, into a mouse in which local neuroprecursors are labeled in green, and in these mice you induce a little injury at the cortex level, what is going to happen is that you'll see migration of label cells that indeed are not the pink one, therefore are not the mesenchymal one, but local precursors that tend to migrate toward the injury, where are recruited, and they acquire a phenotype that looks like neural cells. But as I said, in the early 2000s, I was a neuroimmunologist with very little experience in the field of stem cells, and what struck my attention was this paper that was published in Blood from the group of Carmelo Carlostella from Milan, showing then that human bone marrow-derived stromal cells can suppress T-cell proliferation. At that time, I was still playing, and I still do that, with experimental models of autoimmunity, and mainly with experimental autoimmune encephalomyelitis. And I had, I have to say, the interesting idea that cells that have the potential of stem cells, and therefore they could theoretically repair tissues, at the same time having also the potentiality of inhibit T-cell proliferation, may have been the ideal source of cells for the treatment of an autoimmune disease such as EAE and possibly multiple sclerosis, because indeed, as far as we know, in this disease, T-cell activation and B-cell activation and other immune cells activation leads to myelin damage, as you heard these days, and final destruction, and therefore neurodegeneration. So here is a post-hoc recapitulation of the roadmap that led from the very beginning to demonstrating that MSCs can indeed modulate different aspects of the immune response, allow, based on this evidence, demonstrating that MSCs can treat experimental autoimmune encephalomyelitis, to then further dissect the mechanism of action of these effects through different aspects that I will go through in the next slides. And then, if that was the case, and luckily that was the case, to demonstrate that MSCs can be utilized in humans in a safe way, then have enough scientific evidence of such results to generate a consensus about their use in humans, and at the end, has it happened to be the case, to design the ultimate clinical trial. So let's go through all these steps to show you that indeed MSCs can inhibit T-cell proliferation. This paper from Federica in the lab demonstrated that when you challenge T-cell trigger with anti-CD3, anti-CD28, which leads to, as expected, to T-cell proliferation, in the presence of MSCs, you will see a significant inhibition in a dose-dependent manner, which is poorly responsive to IL-2 administration. And this effect is due to the fact that, as demonstrated here by the intracellular detection of Ki67, which is a protein associated with all the different cell cycles of the cell division, excluded the G0 phase, it's indeed due to inhibition of cell division. So basically, the cells are not killed, as you see here, showing that MSCs do not induce apoptosis, but they are inhibited in their ability to enter the cell cycle, and they are freeze in the G0 phase of the cell cycle. More or less at the same time, actually this paper was published before, in collaboration with Vito Pistoia and Anna Corcione, we were able to show that MSCs can also inhibit a number of B cell functions in vitro. In this case, you see that B cell proliferation is inhibited significantly by, in a dose-dependent manner, by mesenchymal stem cells, and this leads to, I apologize because the light is so bright that here you don't see basically the column of the graph, but I can tell you that there is a graph, and you can even look at the paper, but you can just see the dots on top. But nevertheless, what is shown here is that, wait a second, maybe I can decrease the brightness. No, actually what happens is that I don't see here anything, but you still see a very bright screen there. Nevertheless, MSCs can inhibit also the production of all the different subclasses of immunoglobulins, as well as the differentiation to plasma cells. Oh. In a more recent study, Sabrina Chiesa in the lab decided to address the ability of MSCs to interact with dendritic cells. And these experiments were in part confirmatory, as we isolated dendritic cells from the bone marrow of bulb C mice, and we subjected these, we subjected PBMCs, actually monocytes from the bone marrow of these mice to maturation into mature dendritic cells, either in the presence or the absence of mesenchymal stem cells. Or alternatively, we took mature dendritic cells and we activated them either in the presence or in the absence of mesenchymal stem cells. The data I will present you have been, that I will show you, are exclusively showing the effect of the presence of MSCs when we provide them during activation with LPS, but similar results were obtained when MSCs were added into the culture while we were subjecting monocytes into maturation to mature dendritic cells. So in this study, we confirmed what it was already known, that mesenchymal stem cells can impair LPS-induced maturation of dendritic cells, as is shown here by a significant decrease of the upregulation of dendritic cell-specific markers such as CD11C and CD86, costimulatory molecules, MHC class 1 and class 2 molecules, and interestingly, for what I will tell you later on during the presentation, a significant downregulation of receptors such as the chemokine receptor CCR7 or integrins that are associated with the ability of cells to get into the lymph nodes, particularly dendritic cells. In this paper that has been just published in the PNS, we were also able to show that LPS-activated dendritic cells in the presence of mesenchymal stem cells can produce much less IL-12, TNF, but also IL-10, which is supposed to be an anti-inflammatory cytokine. And this inhibition is associated with a significant down regulation in the expression of a number of key regulators such as MAP2 kinase 3 and FKB1, which associate with the activation of dendritic cells and the production of cytokines. And this indeed is, as I said, associated with a significant inhibition of the phosphorylation of intracellular kinases involved in cytokine production. To make sure that indeed this effect was due to the fact that MSCs are able to inhibit the pathway involved with the LPS activation, we first demonstrated, as it was already known, that mesenchymal stem cells have express all the different toll-like receptors involved in the that are important for the signaling through LPS. This was required by one of the reviewers. But more importantly, we were able to demonstrate that this effect does not require cell-to-cell interaction, but is sufficiently mediated by supernatants from mesenchymal stem cells, as is demonstrated here, where the inhibition occurs similarly, regardless of the fact that cells are in contact or are just exposed to mesenchymal stem cells supernatants. Another thing that is very important for the dendritic cells' ability to perform their functions, of course, is the capability of processing antigens. And with the help of antibodies provided to us by Soldano Ferrone in New York, we evaluated the intracellular expression of some molecules such as MB1 and LMP10, which are involved in the immunoproteasome or the proteasome as well inside the cytoplasm of dendritic cells. And as you can see here, the expression of the intracellular expression of LMP10 as well as MB1 is significantly decreased by the exposure of dendritic cells to mesenchymal stem cells, in contrast to what you observe in control disease or control cells. So overall, these results are telling us that MSCs appear to affect the expression, the surface expression of key molecules involved in the ability of dendritic cells to present antigens, both, as I said, at the surface level, but also intracellularly, and at the same time also to inhibit a number of cytokines which are very important for instructing the immune response, particularly T-cell priming. So what we did next was to address whether these in vitro results resulted indeed in an inhibition of disease to present antigens to CD4 and CD8 T cells. So we utilized transgenic T cells that were specific for an ovalbumin peptide. They were either CD4 T cell, transgenic T cells from the O1110, or transgenic CD8 T cells isolated from OT1 that are all specific for the same peptide over ovalbumin. These cells were sorted and labeled with CFSE, and therefore were primed with dendritic cells that were activated with ovalbumin and pulsed with that were activated with LPS and pulsed with ovalbumin, either alone or in the presence of mesenchymal stem cells. And these are the results. And interestingly, we confirm, as expected, that in the presence of mesenchymal stem cells, dendritic cells pulsed with ovalbumin cannot, compared to control group, cannot properly induce T-cell proliferation, as is shown here by CFSE labeling at flow cytometry. And this is a dose-dependent in these T-cell priming experiments. Similarly, we were able also to show that also CD8 transgenic T cells isolated from OT1 mice were significantly inhibited in their proliferation when DCs were exposed to mesenchymal stem cells in vitro compared to controls. So somehow, a few years ago, we were able to provide an attempt to a tentative cartoon showing how mesenchymal stem cells can affect different immune responses, particularly for what concerns their ability to inhibit the dendritic cells maturation, leading to an accumulation of immature dendritic cells. This will eventually lead to a defective antigen presentation to CD4 and CD8 T cells. But as I showed you before, MSCs do not only have an effect on the acquired immunity through an inhibition of mature dendritic cells and therefore accumulation of telurogenic disease, but also have a direct effect through the inhibition of proliferation of CD4 and CD8 T cells and also an inhibition of proliferation of B cells. And we can also, although this is still yet unproven formally, we can predict that the inhibited T cells are likely to be unable to provide help to B cells, and as you know, B cells need T cell help to produce antibodies. So altogether, this is a quite complex interplay in which MSCs act both on the innate side of immunity and on the adaptive side of the immunity, and at the same time, this effect is probably tightly connected. So how can we transfer this relatively large amount of in vitro studies into a clinical translation? Of course, to do that, we first have to challenge it into the proper animal models. And being a neuroimmunologist interested in multiple sclerosis, we choose experimental autoimmune encephalomyelitis, which is a prototypical, although not perfect, model for autoimmunity. And in 2005, I'm proud to say we were the first ones to show that when, once again here, you need to imagine the curves, but when we injected intravenously at disease onset or at the peak of disease, mesenchymal stem cells into animals with EAE, we obtain a significant amelioration. Here we are basically missing, there is up here a lighter gray curve, which is the control that, please believe me, is there. And interestingly, this effect was not observed when mesenchymal stem cells were intravenously injected at later phases of disease, suggesting that indeed we need to impact the acute onset of the immune response to be able to obtain clinical results, which were not detected when this was when the injections was performed at later phases, when most likely an irreversible disability or an irreversible damage of the central nervous system was already occurred. I think that the most exciting part that that made this paper the most quoted, the most cited in the literature in this field, was that for the first time we were able to show that this effect was at least partially due to the ability of mesenchymal stem cells to induce in vivo tolerance. And how this was demonstrated? Quite easily, isolating T cells from the lymph nodes and the spleen of mice treated with mesenchymal stem cells or controls, and re-challenge these cells with myelin oligodendrocyte glycoprotein, which is the antigen we utilize to induce EAE. And as expected and as well known, control mice have a high number of precursor cells that are specific for myelin oligodendrocyte glycoprotein because have been primed following MOG induction, and as expected, these cells proliferate. But those cells isolated from the spleen and lymph nodes of mice that were treated intravenously with MSCs were not responding anymore, demonstrating that indeed T cells from animals treated with MSCs cannot respond anymore to the immunizing myelin antigen. Later on, we confirmed these results in a different model, which is still an EAE model induced with PLP in SJL, which is typically a relapsing remitting model of autoimmunity. In this mouse, we were able to confirm that MSCs can colonize to a certain level the spleen and the lymph nodes of the cervical lymph nodes of the mice, and this was confirmed also by histology. But the most interesting part of the immunological side, immunological section of that study was that for the first time we were able to show that also the in vivo B cell response was inhibited. In this case, we measure the antibody of all the different subclasses of IgG specific for PLP in mice treated with mesenchymal stem cells in white and controls in black. And once again, in the animals treated with MSCs, we could not observe, or we observed a lower levels of pathogenic PLP-specific antibodies. So I think that this data suffice to say that MSCs are a potent immunomodulatory treatment for EAE, but a major question at that time still remained unsolved, which is, are MSCs capable of replace cells, and therefore is this therapeutic effect based on cell replacement? Okay, one of the big issues here is, are these, I mean, there is a constant thought by scientists that MSCs, as many other stem cells, are immunoprivileged. What is the basis for this thinking? Okay, one of the major reasons to believe that is that when we used human MSCs, as this paper published by Jacques Galipeau's group a couple of years ago, or this data produced in our lab, when we use human MSCs, we can still inhibit significantly EAE. Therefore, showing that, at least to a certain level, MSCs can work through across MHC constraints, because you may expect that injected MSCs are rejected, human MSCs are rejected into mice. Is this the case? Yes, it is the case. In fact, in this paper, again from Jacques Galipeau, forget about the details, which is a quite sophisticated study, but what you may just want to take home as a message is that these cells are rejected by MHC class 1 and class 2 mismatched recipient mice, and the clinical effects that was observed in this case is rapidly lost over time. So what we observe in EAE is that human MSCs appear to work, but on the other side we have evidence that they seem to be wiped out following injection. Why they are wiped out? For example, because Lorenzo Moretta there in Genova, actually at the Center of Biomedical Research that Marco nicely quoted, but unfortunately it doesn't exist anymore because of the flooding we had in Genova 10 days ago, and we have over 5 million euros of damage and all the lab is gone. But in that lab at that time, Lorenzo was able to show that MSCs are killed by NK cells, and still in that lab, Federica, in collaboration with Vito Pistoia, was able to show that also gamma delta can lyse mesenchymal stem cells, as long as NK and gamma delta are activated. So it seems that on one side we have a clinical effect when human MSCs are injected. On the other side, we have evidence that after a while these cells are killed and are rejected, mainly due to the capability of cells of the innate immunity to kill them. Another aspect that stays against the ability of these cells to engraft and replace cells is due to the fact that it is well known since early 2000 that MSCs, following intravenous injection, are sequestered and entrapped mainly inside the lungs. This has been addressed by several people in different labs, and depending on the results published in the literature, we go from 80% up to 99% of MSCs entrapped in the lungs once they are injected. And this is telling you indirectly something quite important, that it is quite unlikely that cells that stays for more than 90% in the lungs can somehow provide you such a strong effect when eventually they reach the central nervous system, particularly because once we work, if we work in xenogenic or allogenic conditions, they are rejected, as you saw in the previous slides. So if we inject it intravenously, most of the cells are entrapped into the lungs. So many people, particularly those interested in the clinical translation of this data, suggested, why don't you inject it intrathecally? So you do what you would do to a human, to an MS patient. Why don't you inject it through lumbar puncture inside the cerebrospinal fluid in the case of humans, and in this case were injected intrathecally through the injection of a catheter into the ventricles of mice. And Sarah in the lab was able to address this question, injecting MSCs either intravenously or intracisternally, and compare this effect with mice, control mice injected with PBS intraventricularly, intravenously or intracisternally. And as you can see here, both treatments were effective compared to controls, but we could not observe any evidence of difference between the two different routes of administration, suggesting that, at least from a clinical point of view, intracisternal and intravenous injections appear to be equally effective. Similarly, we look at histological scores, and also when we look at demyelination, it seemed that both treatments were effective, but did not result in any significant difference. Of course, we addressed the possibility that there is a higher engraftment when we injected cells locally, intracisternally, and as expected, after 24 hours, the number of labeled cells into the central nervous system of mice injected intracisternally is certainly higher than those injected intravenously. But I apologize, here I don't see that this is after 24 hours and this is after 40 days. But as you can see, after 40 days, the total number of cells, despite being higher in intracisternally injected mice, is significantly vanishing, as you can see here, over 150 cells, and here a little bit more than 20 cells as an average. And therefore, this suggested that despite a slightly more effective engraftment when cells are injected intracisternally, so locally, we really do not achieve any evidence of better clinical efficacy or histological scores. And what was interesting to us was the demonstration that even intravenous administration not only is able to modulate immune responses in the periphery, but this was observed also when we isolated T cells from the brain of mice that were either treated intravenously or intracisternally, and look at the number of Tregs, as demonstrated here by intracellular staining for FOXP3. And in this case, we demonstrated that MSC treatment was able to expand the population of Tregs in the brain of mice treated either intravenously or intracisternally. And what was particularly interesting was that these cells appear to produce IL-17. And this is in line with a recent paper that was published in Journal of Immunology showing that MSCs appear to be able to induce a functional change of the phenotype of encephalotogenic Th17 cells into regulatory T cells. So, taken together these results, we, at least I hope I was able to provide you evidence that MSCs can, on one side, poorly enter the CNS, but are still effective as much as are those that are injected intracisternally. And when we injected intravenously these cells, we could tell that a small number was able to reach the CNS, but when we attempted to colocalize GFP-labeled MSCs with markers of oligos or astrocytes or neurons, we still could not observe any significant evidence of colocalization, suggesting that, at least in our hands, but I have to say that at this point in almost everybody else's hands, there is no significant evidence of transdifferentiation. Is this meaning that MSCs do not protect in vivo neurons? No, it's not the case, because when we look at the axonal density of mice treated with MSCs or controls, we observe that axons are much more preserved in animals treated with MSCs compared to controls. And when we look at the number of MAP2 positive neurons in the spinal cord of mice treated with MSCs compared to controls, we once again observe much higher number in the treated mice compared to controls. What could be the reason for this protection in vivo? And a couple of years ago, we were able to show that this effect is at least in part due to the ability of MSCs to act on the oxidative stress-associated response, as is demonstrated here by the demonstration that the upregulation that we observe in the central nervous system, this is homogenate CNS, of enzymes associated with oxidative stress, such as CAT, SOD, or glutathione transferase, which is significantly upregulated at disease onset, at the peak of disease, is suddenly reverted by the administration of MSCs. And this occurs for all these enzymes, as it is occurring also for metallothionein. Metallothionein are oxidative stress-associated proteins that are once again associated with stress responses, and these proteins are upregulated at disease onset, peak of disease, and suddenly reverted by the injection of MSCs. What was particularly interesting to us was that these in vivo studies were confirmed when we utilized neurons, and we challenged these neurons with H2O2, oxide peroxide, that is, of course, an oxidative stimulus. And we observed that the neurons, as expected, upregulate metallothionein, SOD, and CAT, and this was significantly and suddenly reverted by the presence of MSCs, suggesting once again that the in vivo effect we observed following intravenous injection of MSCs is due very likely to the ability of these cells to inhibit the upregulation of these molecules in neurons. Then we move to another aspect that is very much involved in progression and pathogenesis of multiple sclerosis, which is the gliotic response, which leads to the formation of the glial scar and microglial activation. And Simona in the lab was able to easily show that in mice treated with MSCs, we have a significant inhibition of GFP-positive astrocytes, as well as an inhibition of the IB4 expression on activated microglia. And this indeed suggested us to address the ability of MSCs to interact with microglia, which is a cells at the interface between immunity and neuroscience, due to the ability of these cells to play a major role in the maintenance of the homeostasis within the central nervous system, and play a major role as a macrophage-like cells in the brain. In order to address this, we utilize microglial cells, an immortalized line that was triggered with LPS. This line, of course, expressed TLR4, and we look at the effect of the co-culture with mesenchymal stem cells when microglia is activated with LPS. And interestingly, MSCs appear to significantly inhibit the upregulation of key molecules involved in inflammation, such as INOS, TNF, but at the same time, and we will discuss these results later, appear to enhance the production of IL-1 beta, which is in many cases considered a pro-inflammatory cytokine. At the same time, as expected, stress-associated proteins such as metallothioneins, hemioxygenase, or PARP is once again inhibited by the presence of MSCs compared to controls. What was interesting that not only MSCs appear to inhibit the secretion of pro-inflammatory cytokines or pro-inflammatory molecules, but appear to switch the ability of the microglia to express receptors or molecules which are associated with neuroprotective functions by microglia. CX3CR1 is the receptor for a chemokine previously known as fractalkine, which has been associated, as we will see later on, with a strongly protective effect by microglia. And as you see here, the presence of MSCs appear to enhance the expression of CX3CR1 on microglia. NER1 was recently demonstrated in a beautiful paper published in Cell to be involved in the ability of microglial cells to protect dopaminergic neurons following different inflammatory insults. And as you can see here, once again, microglia have an increased ability to produce NER1 in the presence of mesenchymal stem cells. CD200 receptor, once again, a neuroprotective receptor. IGF-1, and this is interesting because IGF-1 is a neurotrophin which has been demonstrated to be produced in microglia following IL-1 beta stimuli. And this, in our opinion, could explain why IL-1 beta in microglia is enhanced, is overexpressed. In EP2 is a receptor for prostaglandin 2, which is once again an immunomodulatory molecule. So on one side, at least in terms of phenotype and mRNA profile, it seems that MSCs are able to switch the phenotype of microglia from a pro-inflammatory one to a neuroprotective one. Is this affecting functionally microglia functions? I mean, microglia features? Yes, it is affecting microglia functions, as is demonstrated by the fact that in the presence of mesenchymal stem cells, microglia activated with LPS enhance its intracellular calcium concentration, enhance phagocytosis, and enhance TREM2 expression. TREM2 expression is a receptor which is expressed by myeloid cells and which has been associated by Harold Neumann and Marco Colonna as a receptor involved in the beneficial phagocytosis of microglia of myelin debris in the CNS, contributing to CNS homeostasis. So, based on the results we observed following exposure of microglia cells to MSCs, we were triggered by the extreme upregulation of the fractalkine receptor on the surface and at the mRNA level in microglial cells. Why we were struck by these results? Because Richard Ransoff, a few years ago, published in Nature Neuroscience a very interesting paper showing that CX3CL1, namely fractalkine, expressed by neurons, can modulate the activity of CX3CR1 positive microglia, and the disruption of this axis is associated with neurotoxicity in mouse with affected by different mechanisms of neurodegeneration. So we were curious to see whether MSCs can produce fractalkine. In steady-state condition, MSCs appear not to produce or produce very little fractalkine. But once we trigger or we expose MSCs to inflammatory condition, which could be the case in inflamed CNS, we observe rapidly a significant upregulation both at the mRNA and protein level of fractalkine. So we thought, is this key molecules involved in the interactions between MSCs and microglia? We decided to silence fractalkine in microglia, and as we hoped, we observed that the effect observed in the downregulation of TNF is partially reverted, but more importantly, is completely reverted in the case of IL-1 beta, is completely reverted in the case of the fractalkine receptor, NER1, EP2. So once we utilize in these experiments MSC's silence for the fractalkine, we could observe that MSCs were not effective anymore in providing a neuroprotective or anti-inflammatory effects on microglial cells. Not only these results were observed at the phenotypical and mRNA level, but also functionally. Once again, silenced MSCs were not able to enhance the intracellular level of calcium, were unable to enhance phagocytosis or enhance the expression of TREM2, suggesting that indeed MSCs may affect microglia function through the release of fractalkine. And to further prove that this was the case, not only we silenced fractalkine, but we tried to recapitulate all those experiments, not using any more MSCs, but adding exogenous fractalkine in microglia experiments. And once again, the addition of fractalkine partially reverted the upregulation of TNF, significantly enhanced the IL-1 beta, enhanced the expression of the receptor for fractalkine, NEUR1, EP2, and similar results were observed also when we look at the functionality of microglia. So the results of this section of our work is telling us that MSCs in microglia interaction lead to the inhibition of the production of pro-inflammatory molecules and at the same time enhance molecules associated to neuroprotection. Importantly, as it is well known, and there are very nice papers, and there is a recent review in Nature Reviews in Immunology on microglia. They seem to dissociate two different functions of microglia. On one side, those functions involved with protection from microbes. So those functions that seem to be involved in the protection following inflammatory insults that are dampened by the presence of MSCs. But at the same time, they seem to enhance all those functions involved with the homeostasis of CNS, such as phagocytosis, which is involved in the maintenance of a clean and healthy CNS. So we like to, play this little cartoon showing that MSCs can, on one side, inhibit bad microglia and foster the appearance of a good microglia. So if we would like to altogether attempt to design a scenario about MSC's function in terms of neuroprotection, we may believe that MSCs can inhibit activation of microglia and potentially induce the generation of microglia with the neuroprotective abilities. They can inhibit the astrocytic proliferation leading to glial scarring. They can potentially inhibit the apoptosis of damaged neurons, neurons damaged by macrophage or by immune cells. And I didn't address this because we were not directly involved in this, but there are interesting evidence, mainly from Bob Miller, the Cleveland Clinic, and from Ludwig Eigner in Munich, showing that MSCs can, both in vitro and in vivo, enhance oligodendrogenesis, and potentially, as I showed you before, enhance local neurogenesis. So I hope I was able to show you that MSCs can, on one side, modulate pathogenic immune response, on the other side, protect CNS or neural cells through different mechanisms, but I still challenge you with the idea that we went through a significant change or switch of the paradigm that started from the idea that MSCs could transdifferentiate and repair tissues, then with the idea that MSCs get to the CNS and release factors in a paracrine way that can modulate the immunity inside the CNS and the immune responses leading to damage of the CNS, to a different step, which is quite provocative, that is suggesting that MSCs do not even need to get into the central nervous system to do their work. Why this occurs? This paper was published in Cell Stem Cells and is one of the best papers showing that intravenous human MSCs that, as I said, they are rejected after a few days following intravenous injection, can improve myocardial infarction in mice because these cells that are entrapped in the lungs are activated there to secrete anti-inflammatory proteins such as TSG6. In another paper published in Nature Medicine, Eva Mazzei was able to show that bone marrow-derived MSCs once again are entrapped in the lungs, and over there they interact with macrophages, which are reprogrammed in a very similar way to what we observe for microglial cells. So they are induced to switch from an M1 to an M2 phenotype, and this leads to a complete attenuation of sepsis. So this year we were able to provide a new provocative idea suggesting that MSCs can be injected intravenously, and once they reach the lungs, they are interacting with other cells. And it's interesting because I know here there are people very much interested in the interactions between immune cells and the vagus nerve, and we are currently addressing the possibility that vagus is mediating some of the mechanism of action of MSCs on immune cells. And from this station, in part migrate, but in a very limited way, to target organs such as the CNS or lymph nodes or the myocardium, but very much leading to a significant modification of the modification of the environment of the recipient, leading to therapeutic results. And how we previous in vitro very novel, in our opinion, exciting results we obtained in vivo, addressing the effect of mesenchymal stem cells on dendritic cells. In this case, we isolated transgenic T cells from DO11 mice, which are specific for the ovalbumin peptide 323, well, I can see whatever, from an ovalbumin peptide. These cells were isolated and labeled with CFSE, were injected intravenously into syngeneic biopsy mice. So let's picture this phase of the experiment. Here we have labeled naive T cells, which are specific for ovalbumin. They are injected into a naive biopsy mouse that was never challenged with ovalbumin. 24 hours later, activated dendritic cells pulsed with ovalbumin were injected at this concentration subcutaneously into the foot pad of the mice. 3 hours later, mesenchymal stem cells were injected intravenously. So the full picture is T cells specific, transgenic T cells specific for ovalbumin and labeled were injected and are mainly going around this mouse for 24 hours. Then they will eventually be primed by dendritic cells that are pulsed with the antigens for which these cells are specific. And then 3 hours later, MSCs enter the scene. And these are the results. What do you expect when a full-blown immune response occurs? For example, when you are injured in your arm, you will eventually have a lymph node that gets swollen. And this is what occurs because a full-blown immune response occurs because cells start to proliferate, because they are activated by cytokines and by dendritic cells, bringing over there antigens that start the immune reaction. And this is the case also in this mouse. In the control biopsy draining lymph node, we observe a significant swollen which is due to an increased amount of lymph node cells. But in the MSC-treated draining lymph node, this swelling doesn't occur. The number of lymph node cells is decreased, not exclusively as a total cell number, but also for all the different subclasses of immune cells participating into the cytoarchitecture of the lymph node. But what was more interesting was that because we utilized a transgenic T cells that can be identified by flow cytometry, isolating T cells from that lymph node, we were able to see the antigen-specific T cells in that lymph node, and we were able to show that while in the control mouse of albumin-specific T cell proliferate, in the MSC-treated mice, these cells proliferate much less. Not only proliferate much less, but also secrete a lower number of cytokines, such as IL-2 and TNF, suggesting for the very first time that MSCs not only inhibit T cell priming in vitro, but more importantly, they can inhibit T cell priming in vivo. Why this may occur? Maybe because dendritic cells that are inhibited, as we observe in vitro, in their ability to upregulate key receptors such as CCR7 and VLA4, which are key cytokines, key receptors involved in migration into the lymph nodes, are inhibited. That was what we observed in vitro. And when we went to look at CFSC-labeled dendritic cells into the lymph nodes of these mice, we observed that both CCR7 positive dendritic cells and VLA4 positive dendritic cells were less numerous, or were present at lower numbers in the treated mice compared to controls. So this data somehow suggested that dendritic cells cannot enter the lymph nodes and start T cell priming. And to further confirm this, we started a collaboration with Gianmario Sambucetti, which is our nuclear medicine colleague in Genova, and we utilized the same protocol, once again transgenic DO11 of albumin-specific T cells labeled and injected into syngeneic biopsy mice. 24 hours later, of albumin-specific LPS-activated dendritic cells were labeled, but not only were pulsed and activated with LPS, but were labeled with technetium-99, which is able to give us insights about the dynamics of migration of these cells within the first 2 hours following intravenous injection. So these dendritic cells were either exposed in vitro to mesenchymal stem cells, or we once again utilized dendritic cells that were LPS activated and pulsed with of albumin, injected, labeled with technetium-99, and injected into the foot pad subcutaneously of these mice. And 15 minutes later, MSCs were injected. So let's see what happens. And in our opinion, this is extremely exciting. These are the results when we look at numbers of dendritic cells entrapped at the level of at the site of injection. Let's see what occurs for negative controls. In the negative controls, we mean dendritic cells that were injected into a mouse in which no transgenic T cell receptor OVA-specific T cells were previously injected. And as you can see here, the kinetic of release from the site of injection of dendritic cells is following a relatively flat curve. Let's see what occurs when positive control cells are in the positive control group. In the positive control group is the group where dendritic cells are injected subcutaneously into a mouse in which transgenic T cells are present. And as you can see here, dendritic cells seems to move toward the lymph node very rapidly, suggesting that somehow they can sense T cells that are able to recognize the antigens they are carrying on their back. Let's see what occurs when MSCs enter the scene. In the in vitro treated dendritic cells, so dendritic cells that were exposed to mesenchymal stem cells in vitro, we observe a curve that is pretty much similar to what occurs in the negative controls. I like to call these cells the sloppy dendritic cells because they are kind of, not very fast in their ability to move into the lymph nodes. And as you can see here, this is their slope. What happens when we injected mesenchymal stem cells in vivo? This is really exciting. As expected, in the first 15 minutes, they behave as is the case for the positive control, because in this first 15 minutes, DCs enter a scene where there are just T cells circulating, which are specific for ovalbumin. But after a few minutes, mesenchymal stem cells were intravenously injected. The ability of dendritic cells to be released from the site of injection and moving toward the draining lymph nodes is completely abrogated. So it seems that within 5 minutes, MSCs that are injected intravenously, they are entrapped into the lungs, are able to inhibit the ability of disease that are very far away from the lungs, that are entrapped into the subcutaneous tissues of the footpad, are capable of completely abrogating the ability of this disease to move into the draining lymph node. Let's see the same experiment the other way around. Here we are basically measuring the number of cells that have entered the draining lymph node. Once again, negative control move into the draining lymph node according to a relatively flat curve. Positive control, they get into the lymph node very eagerly, very fast. Sloppy disease, which have been exposed to mesenchymal stem cells in vitro, move into the draining lymph node in a very slow way, similar to the negative control. Dendritic cells exposed in vivo to mesenchymal stem cells. Once again, first 20-25 minutes, they behave like the negative control. But 10-15 minutes later, IV MSCs were injected, their ability to enter the lymph nodes is completely abrogated. So it seems that we are really changing the paradigm of effects of mesenchymal stem cells, suggesting that MSCs, to be able to change the immune function, do not need to interact directly with MSCs, with the immune cells, but they can significantly impact the behavior of dendritic cells and their ability to prime T cells, even at a distance, through mechanisms that we are currently attempting to dissect, but that are certainly mediated by, not by local cues, but by effects possibly involving molecules such as cytokines or hormones, or vesicles or exosomes, or mRNA, but possible also nerves. So altogether, we can say that MSCs can inhibit T cell and B cell and macrophage and microglial functions that are involved in the pathogenic immune response leading to CNS damage, but at the same time they can also provide factors that are involved in neuroprotection, leading to rescue of neurons and possibly involved in the promotion of neurogenesis. Marco or Mauro, it was an hour and a half, un'ora e mezza, giusto? Quanto? So I have another 15 minutes, or you are dead? 1 hour. Oh, sorry. Oh, okay. All right. So are these results showing that MSCs are playing a major role both in vitro and in vivo on immune functions? I say yes. There is evidence that neuroprotective effects are good enough to demonstrate a significant therapeutic effect provided by MSCs? We don't know. Let's challenge this hypothesis in another model. Here we are talking about ALS, amyotrophic lateral sclerosis, in transgenic mouse models which resemble familiar ALS, and in which there is no inflammatory responses. In these mice, death occurs within 130, 125, 140 days since from birth. We attempted to challenge the ability of MSCs to impact neurodegeneration, taking the most difficult path. We decided to treat mice after disease onset. The few papers that show that some cell treatment could be effective were injecting MSCs. We're injecting stem cells, because this was performed also with neural stem cells well before disease onset. We wanted to challenge this possibility, mimicking a situation that would occur in humans, in which you will treat patients after disease onset. So we injected MSCs at day 90, about 30 days after disease onset has occurred. And this is a Kaplan-Meier curve showing that MSCs can significantly increase the survival of these mice by about 20 days, which is a huge increase of the survival curve of these mice. Not only these animals survive better, but they also perform in several motor tests much better than the controls. We were able to show that in this animal model, a few cells, but once again, a very few, were able to get into the central nervous system after intravenous injections, both in the spinal cord and in the brain, as you expected, because this is a motor neuron disease at much higher number in the spinal cord. Nevertheless, once again, after 35 days, there is almost no more cells, suggesting that this effect may not be due to the ability of these cells to engraft and transdifferentiate. MSCs could inhibit the ubiquitin accumulation, which is a marker of motor neuron degeneration, as you can see here. They can inhibit astrocytes and microglia proliferation, as we observe in mice. In mice with EAE, and oh my God. And they can also display a similar effect we observe in EAE, showing that they can reverse the upregulation of neurodegeneration-associated stress-associated proteins such as metallothionein. And we were able to show also that MSCs can normalize the release of glutamate, which is of course involved in excitotoxicity, glutamate-mediated, in mice with in transgenic mice with ALS, demonstrating that indeed MSCs can protect excitotoxicity-mediated damage of motor neurons. So let's go to the end. We can say that mesenchymal stem cells can be an effective treatment for multiple sclerosis, but also for other neurological diseases. And we are indeed facing a quite interesting paradox, because those cells that somehow attracted the interest of scientists and clinicians for their probable ability to provide therapeutic effects due to their stemness, meaning their ability to repair tissue through transdifferentiation, apparently frequent to appear to repair tissues without much evidence of either engraftment or differentiation. So MSCs are not only a wonderful model for scientists interested in playing in the lab in vitro and in vivo, but can be already utilized in humans. This is a pioneer work by Katerina LeBlanc that was published in Lancet 2 times. And in this study is the results of the effect of intravenous administration of MSCs in acute graft versus host disease, grade 4. Grade 4 is a fatal type of graft versus host disease, and as you can see here, the Kaplan-Meier curve suggests that MSCs can lead to a protection from death of about half of the patients for the time of follow-up in this study. MSCs can be isolated from patients with multiple sclerosis and appear to have the same phenotype and functional ability as those isolated from healthy individuals. MSCs have been already utilized in small pilot studies where MS patients were treated mainly with the aim of demonstrating their safety, and these are studies published by Yamut in Beirut, and this is a study from Dimitrios Karoussis and Shimon Slavin from Hadassah University in Israel. But despite the fact that small studies have been already published all over the world, we felt like it is necessary for this kind of treatment to convey the efforts of clinicians and scientists interested in demonstrating the efficacy of such a treatment. And with the help of Mark Friedman, we were able to put together almost all the scientists and clinicians interested in this treatment. We established an international mesenchymal stem cell transplantation study group that was able to generate a consensus. We were able to obtain the endorsement of important non-profit association and including the Italian MS Society. This group designed a clinical trial which is going to start now. It's a phase 1-2 clinical trial. I'm proud to say that somehow I am the coordinator of this international phase 2 clinical trial, which is going to involve about 160 patients in more than 20 centers from Canada, United States, and mainly Europe, France, UK, Italy, Spain, Sweden, and Denmark. And I don't think you are interested in the design and the outcomes, but this is a proof of concept study that is mainly interested to demonstrate whether MSCs can provide a sign of biological activity utilizing magnetic resonance imaging outcomes as evidence of activity. And I just want to thank you and thank all the people that somehow helped me in this, and of course all